This paper started with a problem that I had when I was writing a grant proposal. I wanted to create some metagenome-assembled genomes, or MAGs, from marine sediment, but I didn't know how much sequencing effort would be cost-effective to get the number and quality of MAGs I was looking for. I couldn't find a lot of guidance in the literature, so I decided to systematically study the question.
To answer that question, I developed a two-pronged approach. First, I thought about the question from a theoretical standpoint. How can we probabilistically predict sequencing coverage in complex communities as a function of sequencing effort? One idea we came up with was to treat individual k-mers and genomes as marbles in a bag and ask the question, how many times do we need to grab a marble from that bag before we have grabbed one of each marble? Utilizing this framework, we characterized the impact of community structure on sequencing effort. Second, we took a more experimental approach. How can we relate our theoretical model to actual metagenomes from different environments? Utilizing our theoretical model, we could predict whether a genome of a certain rareness would be sequenced based on a fixed target coverage and assumed genome size. Reevaluating MAG retrieval as a function of genome rareness created a different picture where it appeared that exponentially more MAGs are being discovered with increasing rareness.
Finally, in order for these results to be more generally available, we created a software tool called GRASE, which uses the results of the theoretical study to estimate the number of sequence reads necessary to obtain MAGs as a function of the abundance of the target organism and a few other parameters. We think of this tool as semi-quantitative, in the sense that the actual sequencing effort to achieve a given result will depend on the bioinformatic pipeline. But we think that this tool captures the relationship between community structure and the sequencing effort required pretty accurately. We hope that this paper and the associated tool will help researchers allocate their sequencing effort more efficiently, so that we can do as much science as possible using our limited resources.